Yes. Awesome. Thank you, Kevin. Okay, so welcome to Frugal Science. I saw a few messages here and there in terms of engaging in projects. So I think I would say accelerate a lot of that right now because now would be the time you should start thinking about projects per se. Uh, the first kind of a way to anchor in projects is through the assignment. And I'm wondering if anybody has done the assignment that we talked about last lecture and is excited about sharing what you found. So just raise of hands, write your name in the chat. That way, I can start picking on people. And the goal is, don't worry about it being perfect. I'm much more interested in all of us hearing about compelling numbers, surprises. Sometimes you might have felt, I thought about this problem and it turned out it was not as big as I thought, or it was much more complex. So the purpose of this is really iterative. You won't be done with this sheet. So I see Danny. Next is Shivam, and I see Debojit, and then I see Ragud. Anybody else is excited? Maybe we'll cover one more. Uh, Anybody else wants to? And Kamal. Okay, Kamal. Uh, Danny, go ahead. Tell
Yeah,
us
so how
what you
much
found.
like numbers wise, do you want me to like really get into it or just kind of my
We'll
overall
see.
project?
I'll leave this as I'll do it as a discussion. So as you start diving in, I might give you more time or less time, depend. Just share. Uh, Say you have 3 minutes.
Okay. So
I
I was really
it's
you've heard
funny
me talk in
because it
class.
was Father's Day. My kids gave me this thing called the toilet timer. So I actually don't know how much time it has. This is uh, it basically starts now. So your timer started.
So in class, you've heard me talk about coral reefs a lot. And of note, I've mentioned this before, but corals, when you think of colorful corals from Finding Nemo, the color actually is not from the coral itself, but rather from small symbiotic algae that are living in the coral's tissues. When the water gets too hot, the symbiosis fails, the corals kick out the symbiotic algae, they turn white and begin to die because they lose their nutrients. And I'm really fascinated in these symbionts, which are called zooxanthellae. And the family problem that I wanted to go after was calculating the amount of symbionts in the world's coral. So if all the world's coral reefs were to be bleached, how many symbionts would be released in the environment?
Hmm.
And I didn't know how much starting information we were allowed to use. So the only information I used were the size of the symbionts and then
Hmm.
just a rough estimate of the amount of coral in the world in shallow reefs. There's deep sea corals, but I just worried about shallow reefs.
Hmm.
So across,
And I
there's a
think,
paper that
Danny,
just came
just to
out.
answer the question that you just said, uh, the information you're allowed to use is dependent on the class of problem. You can use lots of pieces of information. Sometimes it's actually just build your estimate and then go back and check whether your
Mhm.
numbers match the information that was also present. So, because it's a learning tool, it's actually useful to also calibrate sometimes certain things. Okay, go ahead.
So the paper, funny enough, I actually read a paper before we even did the Fermi problem, but the paper had said that they thought there was 350,000 square kilometers of corals in the world. And so that was roughly, I was looking just for other estimates, just to wrap my head around that. That's about the exact same size of Germany. So the entire country of Germany is all the world's coral. And then to go then completely other opposite scale, a symbiont is roughly a sphere with a radius of 5 microns, or my diameter 10 microns. And from there, I then was calculating, okay, if it's a sphere, that means that it's got roughly 500 cubic microns if its diameter is 5. And then I wanted to see how much that would fit into just a square centimeter of coral tissues, just a small little bit of coral tissue. And I was roughly getting around the side. That's 10 to the 6 symbionts.
Mhm.
However, coral's tissue, you can't treat it 2D. So it's actually
Mhm.
it's a slight 3D of tissue around the whole coral. So therefore, there's actually multiple layers of symbionts. But the problem that I ran into was they're photosynthetic, meaning they need sunlight. So likely they wouldn't be right on top of each other. So although it's
Mhm.
3D, I was assuming that you could actually almost treat it in a 2D space, given that they would all fill the same area. So I actually treated the tissue as a 2D space. And then from 1 centimeter, I calculated roughly 10 to the 5 with spacing of 10 different layers. So I had 10 to the 6 per little square centimeter. And then multiplying that over 350,000 kilometers, I went to then just kilometers to meters to centimeters. So I went all the way up. And
Mhm.
I was looking at 10 to the 19 symbionts in just 2D space. So just 2D space, if you take Germany, 2D space, 10 to the 19. But corals are not 2D. The shapes of corals are not 2D. They're all very vast and very kind of crazy. But with not great of a math background, I decided to look at some photos of actually looks like they're almost basketball size shaped of these corals are almost kind of like
Mhm.
spheres, but they're half spheres.
Mhm.
So I then treated this 2D space as basketball sized half spheres. And so then I was calculating what is the volume or what's the surface area that a sphere or that a circle, if you
Mhm.
turn it into a sphere, what would that take over? So I calculated and was comparing if each was a hemisphere with a diameter of 30 centimeters, then I was calculating the area of 1,500 centimeters squared of the surface area, and then compared that, and then now I only doubles. So then I had 7 times 10 to the 19.
Mhm.
But last thing, like I guess something complicated, corals are not only just a single sphere. There's a lot of kind of cones and different shapes. So it's called branching coral that'll come off of it. Think of it like a tree.
Mhm.
So then I was kind of drew a little terrible photo, but it's kind of like a basketball with like little spikes coming off of it. So then I was calculating if they were cones of the
Mhm.
same of roughly, I had 10 centimeters with a little radius, what would that take
Mhm.
up? And so then I calculated the next volume if they were all cones, and I landed roughly on 10 to the 20. 8
Mhm.
times 10 to the 20 symbionts in all the world's coral.
Mhm.
And just
Okay,
because
so go ahead.
Oh, then I had a fun thing. If I was curious if a group were to create the perfect symbiont, the perfect single symbiont that was unbleachable, perfect photosynthetic capacity, how long would it take that single symbiont to grow into a liquid culture enough to repopulate all the world's corals? And through a
Mhm.
quick n to the x, or two to the n, I said that in less than 7 months, a single symbiont could get over 10 to the 20.
Mhm. Mhm. So that's awesome. I think I love this because for a couple of reasons. The first surprise you gave me was just this notion that the all the world's coral are size of Germany. I don't know how many of you knew this, or just intuitively, I had thought that there is more coral on the planet. And it came as a surprise. So this is when you do the numbers, suddenly you realize that, oh, wait a second, that we can bleach pretty quickly.
Mhm.
And it sort of sets the stage for thinking about the urgency in how people talk about it. Uh, the next bigger thread, and I think starting to link it with the project as you guys are structuring your question. So, say for a single clone to reach, one thread that would actually be valuable is for any services that are built around maintaining coral, doing diagnostics on coral, possibly repopulation on coral. It would be valuable to now think about the fact that it would have been awesome if all coral was at one place. Maybe it would not have been awesome. It's distributed, and so there is this energy cost. There is this sort of a cost of how you get a solution in communities and how scalable that solution would be. And so starting to do some now the next thing you should do in this space would be do another calculation to see if what would it take either a citizen scientist or a scientist to reach and report on all the corals in the world, say on a 6 month basis. I mean, just use some kind of numbers. If we were to assume this was the most precious resource in the world and it needs to be monitored, some of that, and the direction I'm going is we might realize, oh, it has to be automated. Suddenly, the sets of solution space that we are in is some aspect of it has to be automated. Otherwise, we won't even get there. Like So what would it take as a capacity? How many number of people would have to be involved in a volunteer snorkeling diving workforce that would go and report every 6 months on health of a coral, for example? Another kind of a diversion that you can take is around dispersion. And this is a very complex one. This is a kind of a scientific problem we work on, is that if a certain set of a symbiont or a genetically modified organism is released in the ocean, what does its dispersal actually look like? Do we have capacity? Do we have the right technical knowledge to think about dispersal in the ocean? Especially, there would be a regulatory aspect around it to say, oh, some countries are okay with modified coral, the others are not. So, trying to estimate and say, oh, if we were to introduce something, what should be the cross-sectional area that you should think about natural propagation? Because coral larvae move, and actually they pick up symbiont in their larval stage. And actually, since you anchored it in coral today, I will show you guys some coral data. It'll be fun. Okay, so this was fantastic. It also kind of immediately introduces the overall question that Danny has been talking about for the last couple of meetings around coral restorations, and it would be phenomenal. And to a certain extent, don't hone in on the solution as yet, but the team should be built around the problem. Although some of you would have brilliant ideas of solutions, don't jump into solutions as yet. When you gather a team with a common goal of excited about a problem, uh, solutions will come. That way, it kind of gives equal footing to everybody. Somebody might have thought about a solution for a long while others will come with broader perspectives. Okay, maybe we're going to do a couple more of these. So let's go. And then people should feel free to post their homework assignment in Discord and Notion. That's the whole point of this. Let others do the same calculation via completely different means or redo the calculation or provide the comments. Remember the and culture rather than the but culture. So I don't want anybody to take a red pen and mark somebody else's homework. That's not what you're doing. But you're adding to it. And if you can add gracefully and add something new, ideas to that, then please go ahead and mark it all up. Okay, next I have Shivam. Then I also have Ragad and then Debujit and then Kamal. Go ahead, Shivam. Uh,
Yeah. All right. So just
but you have to be really fast. I'm starting your
yes.
toilet timer now.
Right. Right. All right. Okay. So I'll just quickly show you the sheet. So I started learning for the tool terms. So I started very simple problem. Number of hair in my head.
Mhm.
So if you can see. So I started with one
I think it's
centimeter.
hard to see, Shivam. Just talk through it.
All right.
It's much
Sure.
more fun to see you talk.
All right.
Keep the paper in front of you and just talk.
Sure. Sure. So I started with estimation, two, three things I had in my mind that I will, since I'm learning, so I'll keep it things simple. So I started with the number of hair in my head. So I started with dividing. So I did my partition of my head, like hair portion, into four parts, and then
Mhm,
I roughly estimated in my head like how much one fourth of that part might look like. Before
mhm,
that, I thought of 1 centimeter square, how much hair on an average in my hair would be there in that. So that
mhm,
now again, remember once in a science of stupid class, there was a show that people either overestimate or underestimate, and when you take a average of that, then you actually come up close to the real number.
mhm,
So
mhm.
thinking about that,
So just one important thing, what Shivam just said is sort of this idea of normal distributions. If you were to just average, talk to everybody, ask for estimates, and then look for an average number between that because we all have this tendency of being around normal distribution. There's lots of papers written on this idea. Some of them are good, some of them are really bad. So I think it's a good starting point, but one thing that would be valuable is also validating it because all people might be biased and the mean might be biased. Okay, go
yeah.
ahead.
All right. So 1 centimeter square. Now my minimum or like underestimate was 100 hair per centimeter square, and maximum or overestimate was 200. Then the entire portion was divided in one fourth. So roughly in my head, I tried to think of graph paper, the 1 centimeter square graph paper, and I roughly in my head estimated underestimate was it would have been around 50 centimeters square, and maximum was 100 centimeters square. So now
Mhm,
there are four possibilities with 100 minimum 100 here 1 centimeter square and
mhm.
maximum 200
Yeah,
minimum.
no, I get the point. Yeah.
Uh-huh.
Yeah. What's
So
the final number you arrived at?
so average of all the four was 45,000
Mhm.
here on an average. So minimum
Yeah.
was
Okay, hold
2,000.
on. So wait right there. Wait right there. How many people here, without knowing anything else, without having done this yourself, and Ashish is exempt from this exercise, how many of you believe this number is under, and how many you believe it's right, and how many of you believe it's over? Did you say 50,000 roughly?
Yeah.
Yeah, 50,000. So, raise of arms of people who think this. I mean, this is intuition. It's important to build your intuition. Uh, does that number sound right? Nobody thinks it's right. Okay.
We could
How
do
many of you think that?
over, we could do over, under, and then
Yeah,
right about it.
over, under, and just right. How many people think it's just right? Yeah, just do the thing between the two. I'll look at the science. Okay, everybody thinks. Some people think it's just right. Wow, lots of just rights. This was overestimate. So people are saying it's less. Uh, yeah, I think my intuition is it's quite a huge underestimate. And I'll leave all of you to redo this calculation because we don't have that much time. Uh, here's a fact. Uh, sea otters are known for living in very cold water. Sea otters are just these beautiful animals in Monterey. And for everybody at Stanford, I'm assuming you've seen a sea otter. It's must of this most beautiful, iconic species. We completely wiped them out, primarily because of their fur, because they use hair on their body as a thermal insulation. And it's a remarkable idea because long hair trap air. So when a baby sea otter is born, the mother pulls air inside this mat of hair to provide buoyancy and thermal regulation to the baby. But the buoyancy can only last a few minutes. The mother puffs the air into the mat of hair on the baby, dives in to get the food, say a sea urchin that it's looking for, comes back up and just in time before the baby drowns. It's it's remarkable to watch, and then it puffs it back up again, kind of like our buoyancy jacket. But to be able to do this, sea otters have more than 1 million hair in 1 centimeter square. It's the highest density of hair known in, I think, all of mammals. So just to give you a perspective of 1 million hair follicles and cells per centimeter squares. So
The
it gives
hair
you
or
the
hair
upper bound.
cells?
Say that again.
You mean the like cells which generate hair or the hair itself?
Actual hair. To get this insulation property, you need
I
to
see.
trap a lot of air. You need a lot of surface area, and one
I see.
centimeter square is 1 million. That's why I think you underestimated. But anyway, we'll find out. But we're not sea
Yes.
otters. Okay, I'm gonna
All
go
right.
much faster now. So Debujit, you don't have to describe through your entire calculation, but just give us a broad idea of the problem you worked on. And
So
especially I'll probably prioritize problems that if you have a problem that's specific to an idea or a project that you're thinking about, it might be much more relevant for everybody because this way you get to recruit people to the project. Is that true for you, Dabijit?
yeah, so I will just talk what is it's about. So last year someone talked about microplastics and tires.
Yes.
So I
Yes.
tried to estimate how much microplastic does one F1 risk maybe can generate.
Oh, that's fantastic. Just last night I had a conversation about a F1 Formula, somebody who loves this race, and we literally were just joking about this problem. Okay, go ahead. I love your, you pose the question in a problem, but you also anchored it in a manner that people will care because people care
Yeah,
about
so
watching these
first
races.
thing, yeah, I
Yes,
thought that where does the tire change most frequently? It occurred to me in F1. So maybe it's easier
yes.
to estimate
No,
if I
it's fantastic. No, no,
go
go
there.
ahead with the estimate. Yes.
So our tire loses around 1 to 1.5 kg during its lifespan, and in F1 they have to change tire frequently. So in one stint, around like I'm guessing 1 to 1.5 kg.
Mm-hmm.
So then next, how many pit stops they should make? So maximum three. On an average, I checked it's around two pit stops. And
Mm-hmm.
then that's why then they have to change roughly around 12 tires. And in an F1, there are 10 teams. Each team has got two cars, so 20
Mm-hmm.
cars.
Mm-hmm.
So then 1 to 1.5 kg into two times pit stops and uh, I mean, calculating
Yeah.
all that, it's coming around 240 to 360 kg of microplastics in the micro environment they are releasing in each race.
Yeah. Yeah. So I want to make it a little more complicated and also a little more relevant to what we might. The hard thing, which you should all now try to estimate, is can we estimate the rough size at which the particles are? Because what you really care about is both dispersal and you care about total number of particles released as well. So this volume is a good number. But now, if you were to assume or start estimated and thinking about how could you estimate the size, and I think this is kind of what I'm saying is this is where the calculations have to get technical. Now you have to think about friction. You have to think about the velocity at which, on average, the car is moving, and hence you know the tire velocity on a surface. What would be the particle size? How would you estimate the shredding of the particle without going and measuring it? Of course, one thing you could say, I can just go measure it. You take your full scope, you stick a tape on the back of the car. The moment the car race ends, you would know exactly what average particle size there is. But it would be interesting if you can go and calculate it. You roughly can estimate the temperature of a tire, so you get to do some polymer physics, you get to start thinking about, and it's valuable. You're building a kind of a skill that sometimes you might realize that you don't have as yet, but I think it would be phenomenal if somebody upped the game and actually calculated what is an estimated particle size, total number of particles, and then what does its dispersal look like? So when you make a particle size and you know gravity, you can actually back calculate its residence time in air. And this is why particles below a certain, people call this carbon soot, the category number 1, 2, 3. They're based on their residence time in air. And because of COVID, a lot of work has been done in estimating dispersal, and that dispersal is what's going to drive, say, how many people are breathing those particles. So, Debojit, I love this. Please extend this further. And then again, everybody should post these on Notion. So, Kevin, just everybody's homework, nobody's really checking. But if you really want to learn, make sure you post your homework on Discord. Okay, next we have Raga. Sorry, I'm going a little bit faster because I want to cover a lot of material today.
Okay, I can do this really fast, but basically, I've seen a lot of estimates. So I was focusing on global health. I've seen a lot of estimates on the number of mosquitoes that exist in
Yes.
different areas during different seasons.
I'm so
But
happy that you chose that problem because in my talks, I use a number from an estimate from a friend of mine in Alaska. And I don't know if when I talked about malaria, if I use that or not. So I've always wanted to validate it. So it would be
yeah,
phenomenal to do this properly. Go ahead.
yeah, so I wanted to do a really general estimate of how that number of mosquitoes translates to actual bites in people. So basically, it seems that like the consensus about the number of mosquitoes globally during high season is around 100 trillion on the lower end. So I chose to use the lower end to account for the fact that I'm assuming that like all of the time is high season. So kind of to like balance it out a little bit. And just for fun, if you multiply that by the size, the mass of one mosquito, this would be equivalent to about like 17,000 whales, like blue whales, mass-wise, which was just crazy to me.
Yeah.
And yeah, so if you half that, because only 50% of mosquitoes, the females, actually cause any bites, you get about 50 trillion. And according to the CDC, females only need to bite around every 3 days to sustain themselves and to lay eggs. And so that's one bite every 72 hours. And so if you divide that by, if you do how many bites per mosquito per second, that's like 3 times 10 to the -6. And then if you multiply that by the 50 trillion mosquitoes, that's almost 200 million bites per second globally
That
for
is
mosquitoes,
incredible. That is
which
incredible.
is
Did you say
crazy
200 million bytes per second? So
per
now
second.
let's go further. Let's also account for bytes a human being will get on average in their lifetime. Dipali is
Yeah.
already probably looking around for mosquitoes in her office right now.
Oh my God. Yeah, and I wanted to
Yeah,
look at
so
different species after this as well.
yeah, I think what I would say, this is fantastic. It's very relevant. And again, we will cover and talk about mosquito biology quite a lot in the class in terms of surveillance solutions. It's valuable to also think about this number from a context of uh, how people get immunity for diseases. You know, many a times when people live in many areas, you get sense of immunity around certain sets of diseases. There has been talk and thread really around gene drives. So this is very critical to estimate these types of numbers around gene drives and how they would spread. So, Raghav, what I would say is just go very, and anybody who wants to join teams on doing these scaling numbers. I mean, these are little kind of mini papers in itself. These estimates are very, very important, and we will post them all publicly. So I would say go deeper, do species, do a sense of thinking a little bit about what does the human intersection to this number actually look like. Challenge your assumption around densities to get a sense, because there are certain areas where there's very high density and low density. So what does fluctuations in this number would look like for, and we know that's there because we experience these fluctuations. Uh, and then
Mhm.
the other part of this story would also be is it turns out mosquitoes don't fly that much. So in their lifetime, they will only go around a mile or two, which came as a big surprise. But and again, it goes back to the exact same idea that Danny was talking about around dispersal of species. Uh, so it has been a paradox right now of how mosquitoes spread across massive places, and especially in the summer. So Mali is a great example. You go to Mali in the summer, it's so hot that you can cook an egg anywhere, and almost all of these sets of mosquitoes die, they disappear. But the rainy season comes back and the mosquitoes come back. And it's actually been a big puzzle in places around the world is how do with seasonality, why doesn't some mosquito species, when a season is unviable, and I know Rena has been thinking about this, how does it come back? And there is a wild idea that's out there which has actually proof. There was a Nature paper 2 years ago from Tolly's lab at NIH, speaking that we should fund NIH. It's a brilliant idea. He put weather balloons in the sky and he demonstrated that mosquitoes are taking these air superhighways a couple kilometers above our heads to disperse themselves. So think about a single mosquito rising in these tornadoes and currents, going a kilometer, 2 kilometer above, passively dispersing and then landing back again on land. And that's, I mean, so connect your estimates with that as a calculation. Take a look at their mosquito distribution as a function of height. They have some numbers in that paper itself. Um, okay, I think we're
You
going to
have
run out of time.
the
Let's
paper or
do,
something?
say that again. I'll post
Sorry,
on
I was just
Discord.
asking if you don't. Okay.
Yeah. Uh, Tozy, just do NIH mosquitoes in the sky. You're gonna get that. Yeah. Okay, Rena, do you want to just do a quick one? Just share your problem per se and maybe the final number that you got, not the methodology.
So I was trying to estimate how many students in India end up giving up STEM after the ninth grade, because just one bit of background is in the ninth grade in India, you end up choosing between different tracks. So like most people end up choosing between science and commerce for some reason, because that's how it's aggregated in the national curriculum. So based on some math I did looking at census data, government data, I realized that approximately somewhere around like 13.5 million students end up stop pursuing STEM every year in the after the ninth grade in India. And yeah, I'm happy to share the full calculation on the Notion page.
Rana, I love this. There is a ton of very education heavy and education minded people in the group. My intuition is that sounds low. I would have thought it should be higher because
That's on an annual basis, like for ninth grade specifically.
I see. Yeah. Yeah, I think one thread to do would be is if you want to dig deeper in this problem, try to anchor it also on specific states. It might be interesting to be able to compare, say, UP, Assam, and say Maharashtra to kind of get a flavor for, is there something that somebody does different that changes? So just add more nuance to it, because this is a really interesting one in terms of understanding kind of what has been often called in science kind of the pipeline of science and where do we lose people. Okay, I'm gonna take one more. Maybe Kamal, go ahead.
Yeah, I just
I
quickly
have to charge my computer. I am still listening, but I'm just gonna go back in my
that's okay.
bag and dig my power. So I'm listening. Go ahead.
Yeah, I just will quickly tell like maybe 2 minutes. I wanted to, so I have been very interested in looking at what is the impact of loneliness across the globe, and we are already in the middle of a lonely epidemic. And so one thing that has always been exciting to my mind is to think of ways by which I could gather more interest in the field, which is not so much according to me so far. And so one estimate that I wanted to think of is what is the economic cost of loneliness across the globe, and how much does it actually cost us while we don't really care to or seem to care enough about it. And so I came at a number which was close to $360 billion a year. This was only for the US because of the way the data I could scrape the data only for the US. But interesting thing is you spend almost 350 or 360 billion dollars per year for all dementia related problems in the US, and that's such a big global concern where a lot of labs are actively putting in so much effort, while nobody seems to care about loneliness as much because people are like, just, I mean, it's a passive problem. It's not something that people are looking at actively. So I just wanted to like use this economic burden to sort of bring some traction to the problem in my mind and maybe talk about it like if I discuss this somewhere. Yeah.
Yeah, I think so. Kamal, one thing you should do is, of course, write up this and then let's all collectively work on this as a write up. It would be valuable because now we have so much massive data around internet use. And I'm wondering whether just, I mean, the amount of data we're generating, like we're online right now. Every button you click, everything is being measured, by the way. So if anybody is clicking on Facebook and Instagram, everything is being measured. I'm wondering whether that massive data stream and there is a way to utilize that data stream to really also be thinking about both kinds of surveys, estimates, and what kind of information can you use to mirror and match. I mean, the more amount of time people spend online, the more you are not with people. I mean, that's the thesis I'm going by. That's a
No, I
hypothesis.
totally agree, because that's the thing that I also mentioned when I was introducing the problem that I'm interested in, because I feel like the more the technology that we are engrossed with and the less we go out, that has a direct impact on how lonely we feel, and so many problems just are a spin or a twist to that sort of a direction, I feel.
Yeah, I think so. One way of thinking about this would be try putting this number in place, depending on just number of time or number of hours per day. An average child, say, let's just assume like a 20-year-old child is okay.
Right.
A young 20-year-old spends on their phone across different states. I mean, this number should be available, and that way you can also look through some of this comparative idea. Again, going back, choose your three favorite states. I'll choose Assam, UP, and Maharashtra, like three states that I feel I have spent some time in, and see comparisons in terms of what sets of trends exist. Because it would be
Right.
valuable to anchor this problem in uh, utilizing some data sets that actually exist.
Right.
Okay, all right. So time's up. I know I had promised a few more people to share. Shivam, did you wanted to say something? I'm about to switch context. Go ahead.
Yes. Professor, last question. Am I audible?
Yeah, we can hear you. Go ahead.
So I was saying, Professor, how do we validate our estimates, right? For example, my estimate was under.
Got it. Yeah. How do you validate? First of all, you can't. If you come up with an original idea, you're the first one. And there are sometimes you can validate, but Einstein, when he decided to measure the size of the atom, he came up with 15 different methods to do the same thing. From the blueness of the sky, he estimated the size of an atom. By measuring the viscosity of water, he estimated the size of the atom. From Brownian motion, he estimated the size of the atom. And it was turning out all to be same orders of magnitude. So try attacking a problem from many different scales, and sometimes you'll arrive at exceedingly different numbers. And as you go and grow in a problem, your estimates and your intuition will get better over time. This is not a yes or no answer problem. It's a way to think. It's the process that's much more important. You will arrive at new conclusions, like the F1 race we were talking about. You will arrive at the fact that, oh, maybe what's actually important is we should not be working on pit stops. We should just be changing the roads in a manner that do not cause, I mean, it's a counterintuitive idea. Trains, the reason we have trains is because steel on steel friction is incredibly low. While it still doesn't have the kind of dissipation, it still has enough friction that the tire can roll, it doesn't slip. While on roads, we only focus on grip, we actually don't focus on microplastics. That suddenly leads to a new kind of a design scale. So the purpose of these problems sometimes is to lead you to new spaces of where solution domains might be hidden. So then it becomes super exciting that you're actually thinking about structure of a tire, and your estimates could have been off in certain sets of ways, but it got you in the right path. So think about the process, write down every assumption that you make. Some things you'll be able to validate. Some things are, I mean, everything that you are all discussing are, I would say, cutting edge research questions. It might sound silly that, oh, nobody really knows such a number, but often enough, really, most exciting ideas can be stated in a very simple sentence. So it's, you should not assume that just because you came up with a question that sounds so simple, doesn't mean that somebody's actually done this. That's the whole point of this. I mean, that's the whole point of doing this very, very carefully and bring all your agencies and your capabilities. You can go as deep as you want in something like this. So you want to use all your knowledge to actually tackle this specific estimate. Okay, so I think we're going to switch topics now. I want to cover carbon sequestration because I am going to make a switch after this week from oceans to global health and diagnostics. But I want to cover one very important thing. And again, the choice of case studies I'm using to cover are also just invitations that I would like a few people to build teams around it. So I'm going to spend time today on kind of technical aspects of carbon sequestration, uh, some sets of challenges, and especially carbon sequestration in the ocean, which is kind of the most that I know of and what we work on. And then we will end up a little bit in terms of small discussion on forming teams, because I think it's going to become very urgent, and we have to do that quickly in some way. Otherwise, we're going to run out of time. Uh, so first of all, sorry, last time I had to leave. I forgot to mention it was not a real emergency. It was a fire drill that we have to treat as a real emergency. But we had to show up on deck. But I'm back on land now, which also means that we will resume, starting Friday, we'll resume kind of in-person sessions, and also we'll have a lab this Friday. So I think for physically, people should try to make time. I think Kevin mentioned, at least for some of you, Friday morning, 10 a.m. works, right?
About
Oh,
10:30
Kevin,
or so. Yeah.
10:30. Okay, so just confirm, Kevin, just let me know. I'm going to move my group meeting. And then let's make sure that the teaching space is available on Friday. I had booked some time there, but let's confirm. Yongjin, could you confirm that that time slot is available in the UTL room, the teaching room? Um, and then the other thing I wanted to mention before we dive in is we'll still have a Zoom session for anybody who can attend. And of course, for physical demos itself, we'll still do them on Zoom too. So everybody is welcome. But we'll try doing, I want to start turning certain sets of things into physical threads. So for Friday's session, what I would like to do as a homework is everybody should bring one optical uh, object that you can find around you. So just if you, for 10 seconds, look around you and tell me the closest optical device. Aiden doesn't count because he's going to say my microscope is right here. Actually, that does count. The idea is an optical object that utilizes principles of optics. You have to bring it on Friday, and we're going to dismantle it. So it shouldn't be something that you really, I mean, we should dismantle and we should be able to put it back together. But there are lots of objects that we dismantle and we're not able to put back together. Uh, so try bringing something that you're okay if you destroyed it. You could bring your eyeglasses. I'm looking at Minyan right now. That's the closest optical device that you have. I happen to have this little kind of a photo frame, and it has these really strange optical properties. You know, I can see critical reflections in it. You can bring something very, very simple as well. Uh, right next to me is a full scope. I have my mouse. Most mouse are optical. Bring an old camera, bring those disposable cameras, anything. Uh, if you're adventurous and you really want to go in, bring an old phone. But I think the idea would be is we'll cover certain aspects of how to learn optics uh, with sort of a sense of play and sense of adventure. Often enough, you know, we learn a lot of principles, but we don't look at enough designs and enough solutions that people have already come up with. So make sure you bring something. I think it'll be much more fun. I will bring some objects. So if you don't have something, I'll give you something to play with. But if you bring it, it's much more personal. Okay, so that's the assignment. And I think this is also true for everybody that's joining online. Okay, let's dive into carbon sequestration. Okay, I am here on Discord. Where should the here is Frugal Science announcements 2025. Can you all see this? resources, random. So I think here, one thing that I would like to do is let's create, you all can create channels, but we should create a channel called introduce yourself. So here people should write quick intros, also introduce sets of projects. You can already start adding, there's teach me anything. Kevin already did one. Make a proposal on what you would like to teach. This can be assignment 1 by the numbers. Here you can post your papers, and then Kevin, just kind of intuitively grow these sets of channels so not everybody is in the same.
Okay.
So
Thank you.
here we can do another one, lab 1, bring your own optics to dismantle. Okay, so you get the idea, but let's create space as you need it. Like this is really your space. So try organizing this. Notion is the much more organized space. And I'm not seeing that much chat as yet, so I think it would be really fun. I will open this also to I'll invite all the past Frugal Science alumni, and so they'll start interacting, they'll kind of share sets of ideas and threads. So it would be valuable to kind of build a cohort before I invite them. Okay, I think I want to transition to sort of think a little bit about carbon sequestration now. We'll spend maybe 30, 40 minutes on this. And I'll pose this much more as what we know so far, and also a little bit seed around a couple of projects that I've been thinking about that I would love if there's anybody who's interested in engaging. So we've talked about the ocean quite a lot. I just docked the boat that we were on, Sally Ride. And we tested a new idea on thinking about sequestration, and I'll talk a little bit about that. If some of you are doing more climate oriented by the numbers, it would be awesome for you to do numbers around how much carbon we're emitting, what sets of strategies are out there for sequestration. There's a lot of general information about sequestration in general, especially the direct air capture, much of what happens in soil, which is picking up. And then, of course, another category is utilization of ocean in that sense. And we will primarily focus on ocean and thinking about just what kind of new tools and technologies do we actually need to be able to scale ocean-based carbon sequestration. Now, it's also important to know that just literally last night, X Prize just announced their X Prize winners for carbon removal. I think the first company, and I don't know what their process is, I don't validate any of it, so I have no clue what these companies claim. But I was reading the claim sort of last night. Marty Carbon or something like that was the first grand prize winner, $50 million. That's a lot of money, I would say. So I was tempted for the first time. It was like, oh, these types of prizes are not bad, because if you actually have that kind of finances, you can do and scale projects. But anyway, you should all read, as a little side homework, read X Prize entries, get a sense of what's happening in the market right now, what are the kinds of solutions. And please, some of you should definitely do by the numbers, trying to either prove or disprove many of the ideas that are being proposed in carbon sequestration, whether they make sense. For example, do a one-page calculation on whether it makes sense to do direct carbon capture. And there is a lot of numbers, and people have already posted this, but just you will understand a little bit about dilution of carbon dioxide in the atmosphere, and then how much energy is needed to remove 1 molecule of carbon dioxide. So lots is there in that space. We're going to mostly talk about the ocean. Okay, so just to review, the ocean is big. And then again, anytime people have questions, you should raise your hand, And Kevin, if I don't see it, because now I'm seeing my own screen, you can just halt me that there is a question. Um, the ocean is big, and that's why working in the ocean is hard. We've talked a lot about kind of sets of technologies, uh, but I want to read a passage from Rachel Carson again. I think I talked about her, but if not, I want to read it again because it's written so beautifully, but it kind of summarizes a lot, both that we don't know, but also some of the fundamentals of how carbon sequestration might be working in the ocean. This is from her book I think she has three books in the oceans, and one of them is really the open ocean, the other one is coastal, and the other is deep oceans. The ocean is a place of paradoxes. It is the home of the great white shark, 2,000-pound killer of the seas, and of the 100-foot blue whale, the largest animal that ever lived. It is also the home of living things so small that your two hands might scoop up as many of them as there are stars in the Milky Way. So Rachel Carson knew by the numbers trick. She was doing this and incorporating it in her writings. Uh, so just think about it. Uh, your two hands might scoop up as many of them as there are stars in the Milky Way. That just gives you the density of microbes that exist in a tiny amount of water. And it is because of the flowering of astronomical numbers of these diminutive plants, known as diatoms, that the surface of waters of the ocean are in reality boundless pastures. What she's referring to is kind of the chlorophyll maxima in the ocean and the fact that there is an abundance of chlorophyll. That's why she's calling it plants, diminutive plants, because diatoms and others really act like they're algae and they are floating plants. They're not technically related to plants, they're in a separate portion of the tree of life. Every marine animal, and from the smallest to the sharks and whales, is ultimately dependent for its food upon these microscopic entities of the vegetable life of the ocean. Within their fragile walls, the sea performs a vital alchemy that utilizes the sterile chemical elements dissolved in the water and welds them with the torch of sunlight into the stuff of life. Uh, you know, I if there was poetry, this is kind of poetry in writing, and then she goes on and does this over and over again. What she's talking about here is utilizes sterile chemical elements. That's biomineralization. So she's talking about silica, strontium, opals, calcium carbonate, all of these chemicals, inorganic compounds that organisms use to build structure. And welds is kind of the process of biomineralization. And she talks about torch of sunlight is just the source of energy to build and grow. It's very important. The process of biomineralization in the ocean is very important because it generates a notion of what is called ballast. Ballast is this idea that cells can regulate their density, and we'll talk quite a lot about this. And so when a cell creates biomineralized structures, it can become heavier. And what happens to objects when they become heavy? Anybody wants to shout out? I just want to make sure you guys haven't gone away anywhere. What
They
happens to
sing.
objects?
They sink.
They sink, exactly. What would happen if an object that depends on light, like a diatom, that is using glass to create a shell and hence becoming heavier, what do you think will happen to it? It will go to the deeper parts of the ocean. But interestingly, the deep part of the ocean is dark. But plankton and most things that need light need light. So this is paradoxical. The fact that life kind of manages to just survive and live so they have to maintain their buoyancy really precisely to remain in the zone that's optimal to them. Otherwise, gravity has never been off on the planet. Gravity is always pulling things down. And when gravity is always pulling things down, flux of carbon is moving downwards at all times. because gravity has never been off. Usually, cytoplasm of a cell is 5 to 10% heavier than seawater, so cells have to actively do something to survive in the water column. And the moment they die, they sink to the bottom of the ocean. And then I'll give you a little bit of an evidence for physical evidence for what does that actually look like. But let's keep on reading. Only through this little understood synthesis of proteins, fats, and carbohydrates by myriad plant producers is the mineral wealth of the sea made available to the animal consumers that browse as they float with the currents. So the animal consumers are whales. Everything that you know in the ocean survives on this vegetative life because copepods, all the zooplankton, then eat the phytoplankton. This is the food chain all the way to whales. Whales would not survive if we didn't have something like this. Drifting endlessly midway between the sea of air above and the depths of the abyss below, these strange creatures and the marine inflorescence that sustains them are called plankton the wanderers. So the sea of air is an infinite amount of contact area that the ocean has with air and atmosphere. And so from a carbon sequestration point of view, it's a really remarkable substrate to be thinking about because it has the highest contact area with the atmosphere. Any machine that we build, anything we build, we will never get this much contact area because the ocean, 70% of the surface of our planet is the ocean. So you have a massive amount of contact area. That means that the CO2 concentration in the atmosphere is at equilibrium with the dissolved CO2 in the water. And so if you measure dissolved CO2 on the surface of the water, you're actually getting a good estimate of what the atmospheric CO2 looks like. Except there are few spots in the ocean, and I think I'll get back to it, but I want to mention something about the Indian Ocean and the Arabian Sea. Very little is known about a few spots in the ocean. Ocean does not just have the capacity to absorb CO2, it can also release CO2. That's the scary part. If you were to do by the numbers, and some of you I think should do this, what you would find is there is more than 40 to 50 times the amount of carbon in just the water in the ocean dissolved as compared to in the atmosphere. So it's an order of magnitude higher amount of CO2 is present in its dissolved state. And what happens when you heat up water? If you take your soda, which has dissolved CO2, what happens when you heat up that soda?
It releases the gas.
Releases the gas. That means the ocean are not just source of sequestration for carbon, they're also actually a massive threat if we don't understand it. And it turns out in the Arabian Sea and many other local hotspots, the ocean actually net releases CO2. And this is astronomical amount of CO2 that we as humans would never be able to tackle. So it's a double whammy. When we think about change in temperature, we're also changing the ocean's capacity to dissolve CO2 to begin with. And at that same time, as processes change, it's very possible for many of these water bodies to actually also release CO2. The other part of this is what Rachel Carson is doing here is she's linking somehow the idea that the sea of air above and depth of the abyss below is connected. And then by the connection is through this object that we will talk quite a lot about. It's called the biological carbon pump. There are two carbon pumps in the ocean. There is the solubility carbon pump and the biological carbon pump. We will primarily focus on the biological carbon pump for today. If people are interested in a project in the solubility carbon pump, which is kind of called also the alkalinity enhancement mechanism, I can do a separate kind of a lab session or a separate private session. We primarily work on biological carbon sequestration, so I'll kind of focus on that. But this sinking that we discussed is what connects the air to the abyss. That's an important aspect to understand. Okay, so, I think I often think about the power of writing. And I want to remind all of you to spend at least few minutes to an hour writing every day, because you could learn the capacity to frame an idea in such beautiful words that, we're still talking about these words. I could not imagine somebody writing something more beautiful than this mysterious as a process. So just an encouragement that if you are a scientist or an environmentalist or an activist, words have power. You should spend time writing. So even when you're writing your assignments, when you're writing anything, you can take it as a chore or you can actually enjoy it. And if you write beautifully, other people will talk about it, which is kind of the whole goal of us sharing knowledge. Okay, so I showed you this before. I'm not going to spend too much time on this. Can somebody remind me? Yes, we did see this video before, right? We talked about this before. Okay, so let's, I'm going to keep moving. I think we also talked about this curve, but if one thing that I'll just mention here is uh, people should look at these sort of curves carefully. If you're interested in the land sink, there is a lot of work happening right now on the land sink, including what is called weathering. And secondly, a lot of people talk about growing trees. There is also the microbes. So if some of you, I would be really delighted if you haven't chosen a by the numbers. I would love some of you to work on soil. Estimate the amount of biomass in soil, estimate the amount of microbes in soil, estimate the amount of fungus in soil, explore how soil as a function of depth changes. Uh, Soil is an incredible source for carbon sequestration. Processes for how we till the soil in farming actually release a ton of carbon, which is a problem. So you have to think about different ways of turning over the soil, which we do need to do to grow crops, but it actually kills and releases a ton of carbon that was stored. So there are many of them. We're going to focus on the ocean sink, and just to spend some time on it, the thing that I've marked ocean sink is in that color. The light green color is the land sink, and the light blue is the carbon estimate when you subtract from the top these two numbers. The top chart is how much we put. This is called the carbon budget. And ironically, this carbon budget is not balanced currently. That's our problem. So we have a huge issue in by the numbers that we cannot balance our carbon budget properly. There's a lot written on it. People, if they're excited about some of the climate side of the story, should go back and do these calculations yourself. But there is a lot of work that needs to be done to balance. It's kind of like having a unbalanced chart sheet when you are in a financial crisis. We are in a climate crisis, but we still, our carbon sheet is actually unbalanced. Okay, so uh, one thing to mention this is I'll talk a little bit about tools and especially optical tools for the ocean, but I just want to give you a preview. Uh, we've been thinking about this idea of how could you experience what Rachel Carson was talking about in the beauty that she's talking about through this chasm in the ocean. So if you notice, the painting on the background is actually a painting from a resident artist in my lab, Rebecca. And Rebecca and I had been talking about, I was excited about making an analogy to the ocean and the Grand Canyon. What's interesting in the Grand Canyon is you can see the depths of what happened millions of years ago. And so the question was, what if you could cut a chasm into the ocean and really see these processes that we are talking about across a function of depth? So the big tool that we have been building and thinking about is how do we really build the kind of tools that allow us to understand processes that occur as a function of depth in the ocean? Because everything changes as you go from the top to the bottom. And the video that I'm about to show you is a real-time video of if you were shrunk into a tiny submarine, put in a given depth of the ocean, this is what you would see. And I'll just play it. It's a very short clip, 30 seconds or less. But I want to visually explain to all of you what would you see. So that's a little dinoflagellate that was climbing up on its migration, being harassed by a copepod. Gravity is pointing down. And then you see this massive biomass of diatoms just sinking. I'll play that again. I got this video in a very special place in the English channel, and I'm very excited about and happy about capturing this video because it sort of gives you a sense of what might be happening. Oh, looks like suspect. Okay, so what you saw as a rain is, I mean, I also compare it to the Matrix. It's kind of the Matrix of life, if some of you know that scene from the movie. An incredible biodiversity is literally migrating vertically up and down every night. And migrations have been documented. But what happens is that the biology as a function of depth also changes dramatically. So we do a lot of work on sort of fundamental science of this to try to really understand how can biology survive under high pressure, low temperatures. Much of the expedition right now was around that. And it starts to give you a sense, because I talked about pressure, it's roughly around 1,000 atmosphere in the Marina Trench. So it gets higher and higher pressure every 10 meters that you take. So the biology gets much more extreme as you go further deeper. But to give you a sense of this vertical stratification, let's do a quick story. This is a game that we play, and I'm just gonna I'll show you a video of what we're doing. It might look silly, but we're actually doing a real experiment, and then I'll ask all of you. Everybody has eaten a Skittles or a M&M before? Show of hands, you know what a Skittles or M&M is? The little candy that's the size of a penny but has a color, red, green, blue. Everybody knows that. We are standing on the edge and we are doing an experiment. Everybody is dropping a Skittle or M&M into the water, but they're different colors. And we are watching how many seconds pass until you stop seeing your Skittle. And I'll let this video play. Oh, everybody is focused on their Skittle. Okay, hold on. Okay. Can anybody explain to me why did we did this very important scientific experiment? Why did we drop colored Skittles into the water? What can you learn by measuring the time of when the Skittle will disappear?
Where are you talking about, like as it sinks or as it goes further away?
Sorry, as it sinks. We often try to do it in a quiet ocean.
Light absorption from the ocean as
Mhm.
it sinks, looking at different colors
Mhm.
and trying
Okay.
to identify.
So
Oh,
what Danny's talking about is the idea that as you go deeper into the water absorbs light. And as water absorbs light, certain wavelengths will get absorbed faster than the others. So the deepest wavelength that goes in blue, the fastest that gets absorbed is red. So if you don't even have that wavelength, you cannot detect a color if that wavelength is not there. So it's intuitive if you think about, but this, by using Skittles and dropping more and more Skittles, and because Skittles are just sugar, and I think it's okay, we're not polluting, it's not a piece of plastic. You can measure the average time that your depth when the Skittle disappeared, and you can actually create this curve that gives you a sense of how light absorption changes as a function of depth. And, of course, there are more scientific methods to also measure this, but it's valuable to show that I think it's important to understand that all aspects of the ocean are vertically stratified. The word stratification means that things change as a function of a gradient. And so what's happening here is you can see light. That's why we call it the deep, dark ocean, because no light penetrates there. It's dark out as you go deeper. That's because light is absorbed as a function of time, but its blue goes the deepest while red doesn't penetrate at all. So different wavelengths have different penetration depths. Similarly, you can see the pressure is drawn as an upside triangle. Pressure at the surface of sea is 1 atmosphere by definition, right? I said pressure at the Mariana Trench, which is around 10 to 11 kilometers, is around 10,000, no, 1,000 atmosphere. So that gives you every 10 meters, this is a radial. Nutrients often are in this upward kind of a triangle. Lower nutrients on the surface because lots of organisms, the biomass is very heavy. They consume the nutrients, so the nutrients change as a function of depth. So you can now imagine this becomes a very complex ecosystem. And what we have to figure out is how life organizes along this column to be able to think about naturally how sequestration is happening. And so to give you a context of this, of just how much life are we talking about, this is a video from a dive. This is not my dive, but I've also done blackwater diving sometimes. And this is the density of things that you see. You're just watching a film that somebody put a camera in a bloom essentially in the water. This is not concentrated water. This is literally the density of macroscopic life. We haven't even touched the microscopic life. And it starts daunting on you. What are we talking about? And again, I'm not saying all the ocean is like that. Just like this is the Amazon of the ocean. This is a high bloom. There is also what is the deserts of the ocean, and there is almost nothing. And often enough, as oceanographers, sometimes when we go out and we find nothing, I mean, sometimes we know there's also oxygen minimum zones that are starting to grow now. But it just gives you the challenge of how do you understand this ecosystem to better understand sequestration. So this is just the baseline of biomass being present. Now, the second complication, and again, the first set of people that did this are right here. This is Gloria on the right. She was the safety deck officer when William Beebe and Otis Broughton went down in a tiny little steel ball. I'm actually curious, how many of you raise of hands if given a chance to go in a perfectly safe, modern submersible? How many of you would volunteer to go? Say, go down to 5 kilometers. I want to see. Uh, and it's perfectly okay if you say that, nah, not for me. Uh, Actually, I'm seeing a significant number of hands. That's amazing. This is just like space travel. Now, keep your hands up for a second. How many of you would go if it was just a ball of steel bolted together with a semi-cracked piece of glass that was attached, and there was a single wire that if there is a danger, you shake it a little bit and it reaches the top. And so that's the challenge. Like, think about the psyche of Otis and Beebe to have crawled into that tiny little metal ball. There's almost nothing. They knew, they had done the calculation that this metal ball will not crush. What they did not know is actually whether this glass will break. And in one of the dives, there was also a crack. But Otis was an engineer, and he thought that with pressure, the crack would remain together because it's compressive. He turned out to be right at that time, but there are other modes of glass fracture that occurred that he did not know about. You can see literally it's bolted. There is a cable. And then Gloria, that played a very important role, and very few people know about her. She was the safety deck officer, and she has a telephone line going through this cable, and it's literally in a crackling sound. She would hear what they are describing in the bottom. And she would write in her notes, and she would write everything, and sometimes she would double check because she was not sure the oxygen amount was going down. So now you can remove your hands. I just wanted to get a sense for how many of you would go down. uh, I think it's kind of sometimes this kind of bravery opens up fields in ways that nobody would have thought about. And actually, BB was not even an oceanographer early on. He was a naturalist. He was just so curious of what's down there. Uh, One important thing that he did, and now I'll kind of come really focus on carbon sequestration. Uh, Rachel Carson wrote this later, but it's words that BB had used, not in this exact form, but what he said in this first dive is quite strange. I'm not talking too much about the deep sea fish. I'm happy to talk about that sometime. We also work a lot on how biomass is distributed, but I'm just going to focus on carbon sequestration. The first surprising thing he noticed, he said that it's snowing down here. And Gloria at that time wrote that down, saying, no, these guys either are drunk or there is some hallucinations because they have low oxygen. So who knows? Then she checks. It's like, what do you mean it's snowing down there? She's like, I can see it's snowing down here. And what they meant is this. And I'm just going to show you that snow so you have a sense of uh, what they explicitly experienced. And I really want to show you guys this video. Where is the snow? Okay, I might have removed it. It's snowing. Yes. Okay. Did I show this video before? No. Yes, I did show this video. Okay, I'll just show it again. This is from the Arctic. We take an ROV, we put it into the water. This is what we see. So this is the safety officer. Pranav is in the lab now, is putting the ROV. We're going to put it on the edge. By the way, from Frugal oceanography point of view, for $5,000, now I can buy an underwater drone. Phenomenal as a technology for exploration. If any of you are interested in thinking about some of these sets of ideas, the space of underwater imaging using simple underwater drones has absolutely exploded. Phenomenal. And I think it would be very valuable to also be thinking about what does the design space currently look like? What are the open challenges? But with that as an object, this is what you see. This is what BB was talking about. And what we're doing is we are just lowering it down. Every little puff, it's pointing down and it's going down and going down. And as far as the bottom of the ocean, and this was only 50 meters, so we reached the bottom very quickly. You see all kinds of life forms. This is a fun discovery that we have made about a very high distribution of clonal brittle star species at the bottom of the ocean under the ice. And kind of one of the things that I am sad about is every time we talk about the ocean and we start talking about mining the deep sea, we don't even know which communities live down there. And we are after these nodules, and all of these countries, including India, now has a kind of a thread around one of the marine budget that they included. It sort of says exploration and exploitation of the deep ocean. We don't understand anything about the ecosystems. We don't as yet understand who lives there, what these communities look like, and we are ready to exploit. So anyway, another thing to think about if some of you start doing by the numbers for these nodules and kind of look at its conservation impact and economic impact in some sense. Okay, going back to sequestration, and so I just want to pose this as a challenge. What's really going on is essentially a simple curve like this. So we're going to not focus on, I'll pick up on technologies some other time, but I want you to just focus on this graph on the right. We talk about photosynthesis. We talked about things die, and a lot of things in the ocean, as they die, they sink. And they form what are these called marine snow particles. And these particles, because of gravity, sink to the bottom of the ocean. And because of this natural process, the ocean is locking carbon down below into the sediment for tens to hundreds of thousands of years. This is a process that continues over and over again. It's a very complex process because you saw in a tiny fraction of the ocean, you saw this massive density of snowfall. And to give you, and then Kamal, I'll just take your question in 1 second, but to
Yeah.
give you all the sense of what it means, uh, I want to show you. Some of you have seen that Lord of the Rings scene. Do you all know about the White Cliffs of Dover? Maybe, maybe not. These are mountains in the UK across the English Channel. And you can see these massive cliffs, but they are perfectly white. So a ship for scale. But these mountains are made out of single cells that are 5 microns. This is a massive amount of carbon that got stored in the bottom of the ocean. And our ocean, on average, has roughly half a kilometer of this sediment goop that has been for billions of years a carbon sequestration technology that already works. That's called the biological carbon pump. So all of these sinking things that you see are very good for the ocean and for our atmosphere because they bring food to the deeper ecosystem. That's the primary food in the deep ecosystem. And then the second thing that they do is they check the balance on CO2 concentrations in the atmosphere. If the CO2 concentrations are high, dissolved CO2 is high, biomass is produced, organisms die. As that biomass sinks, you actually get sequestration. Now, the challenge and the technical classical framework, you can start by the numbers. The simplest idea is what is called Stokes' law for sedimentation. If you were to take a penny, I'm actually curious, and you drop it and the ocean is 4 kilometers deep, how long do you think, you can either write in your chat, how long do you think a penny will take to reach the bottom of the ocean? Okay, I'll give you guys options. One option is 1 minute. The second option is 1 hour. The third option is 1 day. And the fourth option is 1 month. And the fifth option is 1 year. You're taking a penny, and for people not in the US, choose your favorite small coin. And I'm actually curious. Maybe people can raise their hands to say 1, 2, 3, 4, 5. I see a lot of fours. This is a biased effect. Okay, 4 was what? 1 month, right? 5 is 1 year, 1 month, 1 day, no, 1 week. Sorry, 1 week, 1 day, 1 minute. 1 week. I'm seeing 1 week to 1 month. So I'm gonna leave this as an exercise actually. Given this formula, this is a little bit of math that you all need to do. Put all these numbers in. You will get a velocity of a sinking object if you account for, and again, a penny doesn't really follow the Stokes law. It's much more complicated, but you can start with something like this and you'll get a rough estimate. Do the density of nickel, do the density of seawater, look at the size of the object. This is the viscosity of water, and then you will get a term that will give you the time. And then do the same thing for a single cell. And you'll realize that there is a lot of residence time. Things do take time to get there. Now, the challenge is because things take time, and especially biological things are much, much, much lighter, because I said the cytoplasmic density of a cell is only 5 to 10%. You get what is classically in carbon sequestration called the Martin's curve. And this is really what I want to end on soon. I know we have a couple of minutes, but can people stay for a few more minutes? Thumbs up if you can stay, unless you have to urgently leave? 5 extra
We have
minutes.
until 12:20.
Okay, okay. So I'll probably end around 12:10. So, and then I will open for questions. I'm rushing through all of this. I love telling stories and I get lost in them. Okay, so what do I mean by Martin's curve? J.H. Martin in 1987 published this remarkable paper. It was a accumulation of, we put in the ocean this thing called the sediment trap. And I'll show you what the sediment trap deployment looks like. We were just doing these. This is called a pit trap. And I'll see if this is supposed to be a video. You see these little bottles? They have an orifice of a given area. Sometimes we put a giant net there. Sometimes we put bottles. And then we let this go at the bottom of the ocean. It keeps going down. It keeps going down. We let the line sink 500 meters, sometimes even a kilometer. And on top of it, we will attach these buoys. So now the object has sunk, but it's attached on a buoy with a flag with a rope attached to it. So you have this hanging thing, which we now call a sediment trap. Again, super easy to make. You can make an object that's going to collect what is falling and sinking in 1 meter square of a radius or this 10 centimeter square of a cup. We leave it for 12 hours, 24 hours unattached to the boat. You can see it has a flag, it has a radio transmitter, we can track its position in GPS. We let go of it, it's freely floating. And then we collect how much biomass we collected in 24 hours. And from that, we make this set of an estimate of what was the flux and total carbon. So you can see these as molecules of carbon per meter square per year that is falling at a given depth. And every one of this dot was you put your sediment trap at a given depth and you start, Martin noticed this really interesting exponential. Somehow the carbon is disappearing. If it was not being consumed, what do you think this shape should look like? Dead stuff sinking to the bottom of the ocean, the shape should look flat. But it's not flat. It's reducing by an exponential. And then it sort of starts to flatten out, but it's still reducing the deeper and deeper it goes. So, quick quiz. Why would it be reducing? Where did the carbon go?
Eaten by
Go ahead,
some
Shiva.
other.
Eaten by some other things. What could be eating carbon as it goes deeper? Sharks? Whales?
Could
What
be.
else? Yeah,
I said could be.
could be. Could be. Remember, people claim this is a universal curve. Organisms consumed. Chemosynthesis is very different. This is organic carbon. It's food. Who lives in the ocean in very high abundance? Remember the Milky Way that in your 2 scoops, you're gonna have a Milky Way of what?
Plankton.
Plankton, that's correct, but very specifically, what kind of plankton?
Diatoms.
Items.
No, diatoms eat light. Diatoms eat light. They don't need to
Zooplankton.
eat this. Huh?
Zooplankton.
Zooplankton. That's one. That's one. You guys are missing a very big category.
Phytoplankton?
Phytoplankton means
Bacteria.
light. Phyto means light. They don't need to
Bacteria.
eat this carbon. Huh?
Bacteria.
Bacteria. Exactly. You know, how many of you study bacteria? At least some of you use bacteria. They rule the world. Actually, both bacteria and archaea. There are incredible species of archaea. We just don't like talking about them. But now, bacteria don't work for us. They do this process called remineralization, because they are eating this carbon and actually they are emitting CO2 back. And bacteria are also small, so when their biomass increases, they don't do sedimentation. Just from Reynolds numbers point of view, they are so small. If you fit their number, you will find that their sedimentation velocity will be infinitely small because they are small. So ironically, we started with very high flux. Bloom happened, you got a lot of carbon, it's dying, it started to go, and along came bacteria and messed up everything, and they pumped the carbon back. So, biological carbon pump, the influx term is gravitation, sinking, and the outflux term that returns that carbon at a very fast rate is what is called remineralization. It's the metabolic processes in bacteria that are now eating this carbon and putting it back up. And the ratio between these two determine how much sequestration you do. So you can have a lot of carbon flux, but that doesn't mean you really stored it. You only store it if you go below 300, 400 meters when that curve starts to flatten out. And I'm now going to show you the state of the art, like some of the best estimates we have. And I want to tell you why this is a very important problem as a class we should handle. A friend of mine compiled all of carbon sequestration data using sedimentation speeds and things that are known. And this is literally the noise in the curve. This is 13 to, I think, maybe 15 different measurements made by people. It's all over the map. You could put any line in here, frankly. And I mean, this is very, very good statistics that's been done, and that's how people estimate kind of rate of sedimentation and new relationships. And I think when we looked at this in the lab, we realized this makes no sense. I mean, this is good start. This is a really good start, but we need to do better. And then we ended up developing an imaging tool that we can now directly watch these things sink at a microscopic level. So I'm going to show you that. First of all, if you don't believe me, read these papers. I'll read the title. I wish I was bold enough to choose titles like this for my papers. One day I will, but I'll read the title. Assessing the apparent imbalance between geochemical and biochemical indicators of meso and bathypelagic biological activity. What the dash is wrong with present calculations of carbon budgets? They're being very honest. You can fit your favorite four-letter word in here. And what they're pointing to is this notion that we still can't damn balance our carbon budget. So this is important. And one of the reasons is just that our measurements are so sparse. Being in the ocean is costly. I've told you this so many times. Instruments are super expensive. Argo float costs $100,000. They sink to the bottom of the ocean after a few years. We still need 100x more increase in infrastructure, 100x, to even be able to measure. Forget about all these people that are talking about sequestration technologies to ever actually be able to then measure and check them to confirm that anybody that claims what they're doing, which is called, the field is called MRV. And what you're trying to do is verify if somebody claims I've come up with a clever trick for sequestration, did you actually do it? So one of the MRV technologies that we developed is a method to directly image. So now, instead of the snow that you saw and around you, you're actually sinking in the frame of reference of this object. And I'll tell you at a later time how we did this. But where's my video? I thought it was a video. Ah, okay. This was the most fun part. Okay, maybe that's the video. Yes. Num num num. Okay, so I'll play this video one more time. What you're seeing is carbon is sinking, and now we can directly measure its velocity. The highest velocities that we measure for this carbon sinking is around 200 meters per day. So I'm already giving you a hint for your penny problem. But by making these measurements, we notice the bacteria are doing something really bad. Bad for us, good for them. They generate this parachute behind this particle, and this parachute essentially slows down sinking. Why do they want to slow down sinking? Because they need this food. They don't want to go deep down into the ocean to eat this food. So they've generated these polysaccharides. We're calling them mucus tails or comet tails. And when you do this calculation, we actually realize that bacteria slow down the biological carbon pump, and the rough estimates of this is around 80 gigaton of an error in our current calculations when we account and don't account in the wrong direction for these parachutes. Now, these are molecular processes, but we know nothing about molecular processes in the ocean. We know nothing about secretion rates. We don't even know what polymers these are. There's a billion types of polymers that are being released. These are also communities. These are living communities. And to give you a sense of the living community, I'll also show you in the lab now. This is work with a postdoc in the lab, Rahul, and many people who've made this tool possible, which we call gravity machine. I'll actually show you why. So now we are not just imaging something, marine snow, we're also watching. I've now turned on another fluorescence channel and we're watching the bacteria. So remember what I told you about bacteria? This is the native density of bacteria in seawater. I'm using a DAPI stain, so this is a nucleic acid stain, so it's staining everything. And you notice some of them just pass by. There's like, no, thank you very much. My belly's full. But look at the other guys. They're like, oh, there is food. And kind of in grad school, this is how I was. When I would see any seminar that had free food, you change your plan. It's like, oh, of course, I'm going to attend that seminar. And so this is that rolling mill. And you would notice that they are all just now hovering around. Also, this entire particle is now chock full of bacteria to begin with. So this is really also the process of remineralization in some sense. So couple of takeaways. One is we're now starting to scale this imaging tool to make more of them so we can directly estimate this reduction in carbon sequestration. But it's leading to now thinking about a low cost verification technology for ocean-based carbon sequestration. So we're now starting to apply all the things that we did in imaging, from foldscopes to planktoscope to gravity machine to squids and octopi. We're taking these microscopes in situ. So it's a little bit of a platform, kind of like Argo Float, but that means we need a cheaper buoyancy engine. What is a buoyancy engine? Buoyancy engine is an electronic gadget, a little bit like a submarine. But what is a minimum lowest cost buoyancy engine? Currently in the market, they cost roughly around $20,000, $25,000. Electronically, I want to be able to, everybody's played that game, Cartesian diver. You know, you change the pressure and it goes up and down. No? Okay. Homework assignment number 2. Very important.
From Marvels.
I'm sorry. I was gonna show you guys a what is a good picture? I think you guys will be able to search for it yourself. Maybe I want these do-it-yourself pictures, not these commercial versions of these. Okay. So when you have a free moment, get a bottle of Coke, follow these instructions of how to make a Cartesian diver, and you will see it's an object that goes up and down as you change pressure. Now you have to think about why, and you should all make it. It's trivial to make, super easy. But what we need is a kind of a buoyancy engine that can be electronically controlled, that can be stable, so you can put instruments on it. Currently, all instruments that are free floating are way too expensive. Another class of problems is really around, what if we understand the biology of these processes? Could we think about enhancing sequestration? Could we think about processes? And again, gets into the tricky business. It has huge ethical issues around us claiming, but unfortunately, we have come at a point where we have to at least think about them. I'm not saying by the act of thinking about it is not doing, but at that same time, it's valuable to just think about where the biggest wins are. Currently, the ocean is the largest gigaton scale sequestration technology. We have been, there was a collaborator in Duke. They came up with a brilliant idea. We're now just supporting and helping them of using natural clays. So, the reason we were on a ship is we actually did this experiment now because I told you about Sahara. The Sahara Desert dumps so much dust into the ocean naturally. It introduces iron. Iron fertilizes is the most necessary mineral that's needed for plankton to grow. So iron fertilization is a big deal. Everybody's talking about it. We don't really understand it that much. But iron fertilization and biomass is not the problem. You can grow a ton of biomass in the ocean. That happens naturally. What you cannot do is sink it because it's microscopic. When a ton of biomass happens, bacteria are super happy. And with this process, we have discovered they are doubly happy because they have evolved to do this for 3 billion years. The biological pump has been working since the time life has existed in the ocean. So there are evolutionary aspects to it. But one of the experiments that we did with Mukul's lab is, and they kind of introduced this technique, we are now trying to understand its biological mechanism, which is if you introduce natural clay, it forms flocks naturally because of charge, because of bacteria on these. You give a substrate, which is these are clay particles that are a few micron, tenths of micron. They don't sink by themselves, but they trigger a nucleation reaction where biomass starts assembling, and then attach with other cells. And what we notice, and we did this in the just literally a week ago, and we can clearly see that in our gravity machine there is like this massive nucleation event that happens, and around these little clay particles bacteria come, then the clay particles grow, then the plankton come. And we only have estimates right now, but it can speed up the process of this sedimentation by almost twice as fast. And the bacteria parachutes that I showed you, it slows down the process by half. So it's a little bit of a counter. And why clay? Because clay is heavy. So you're adding also a density term simultaneously in that equation. And you are making the particle larger. So remember, density is important because it's the sedimentation velocity is the product of density differential, rho, and r square. And the radius is important. So the larger the particle, the faster it will sink, but by an exponential of a square multiplied by the density. So this has a double win. Uh, so anyway, this is one example, but there are lots of examples floating around. In kind of a frugal oceanography space, the two kind of spaces I want us to think about in terms of projects is one is the space of just making instrumentation in oceans much more cheaper. I mean, the OpenCTD was a great example, but we can do much more in that space. Currently, most of those platforms don't exist. The second space is really around these biological ideas. If we understand these processes better, can we actually improve them? Can we enhance them? Can we at least get mechanistically much more detail at a lower cost? Can we understand uh, much of what's happening from measurements point of view? Are there completely orthogonal ways of measuring carbon sequestration without imaging? Uh, electromagnetic measurements, all kinds of sets of things that might survive a lot more. This is a huge space because at the end of the day, it actually requires, uh, we are almost blind to the depths of the ocean, and sequestration doesn't happen on the surface. So satellites and things are good, but they're only very limited. Much of sequestration is happening between 200 meters to 1,000 meters. So we need technologies that actually have to survive. So even if you work in your frugal science project, a cheap, low cost encasing that will not crush itself for say 1,000 atmosphere using low-cost parts, that's a huge boom because anybody can put their precious instruments. Right now, the casings themselves are super expensive. And there was actually a project testing PVC pipes as a way to encasing instruments, and it's gone quite far. I think it can go down to 400 meters now. This is a do-it-yourself high pressure encasing, and in one of the oceanography lab, we'll definitely try building one, and I'll donate my, I believe in the project, so I'll donate my GoPro. We will lock that GoPro in this PVC pipe with some glass, and we'll send it down the ocean somewhere in Monterey uh, to see whether it works. So I think you can start very simply, literally from a PVC pipe. I just want to mention that while oceanography feels super technical, at the end of the day, it's super simple ideas that combine together that scale. The other space is how do you make community fishermen and communities that are already on the water much more tuned to collecting biological data. You know, that's how Planktoscope started, but that's not the end of it. There's a massive opportunities in thinking about people mapping the ocean, and especially deeper ocean. 2 years ago, we had a really cool project that I want to continue. We developed an idea of a passive probe with no electronics. So that's the key, no electronics, that you just drop. It goes all the way to the bottom of the ocean. It opens and collects a tiny bit of sediment because understanding the sediments is very important. So it collects just a small vial of a sediment, and then naturally something happens when it hits the bottom of the ocean, and then it naturally rises up. And then you collect it back and you've got your sample. So if you're on a tiny boat as a fisherman in the Azores Island, which literally are 5 kilometers deep, you can collect sediments in a very simple way. Sediments are important because they give you the flux that's happening, that is actually reaching to the bottom. So, I mean, we did a lot of work on sets of mechanisms, but it's not anywhere close to testing yet. So I'm just giving you another example. It was a really fun way of thinking about how do you give everyday people access to the deep ocean. Okay, I will stop there. Open mic. I know I ran a little bit late, too much to do. I already see Ragad's hand. Uh, Fire away. I still have another 10 minutes, so I'll stay online. Anybody who wants to stay and yap. I know it's pretty late for folks in India, so I apologize for this. I will try to finish on time usually. Okay, so open mic. Anybody who wants to say anything, think with me, just raise your hand. Go ahead, Raghav.
I had two quick questions. The first question was about the clay project you were talking about. So is the objective of it to collect bacteria so that they don't collect on the actual carbon sources, or what's the point?
No, the objective is to use clay to enhance the flux rate down below because
Oh,
the clay help forming the marine snow particle faster. And no matter, even if bacteria are still remineralizing, more is being made. So it increases the production of larger particles. But if we can increase the amount of clay per particle, we are playing with that density term. Remember that rho. So I think, as I promised you, uh, I want you guys to write down this equation and just stare at it long enough, and we can derive it in 5 minutes. Remember this term rho? This is rho total. This is the density of the particle. By adding clay, we are increasing not just, usually the density of these particles is around the cytoplasmic density, which is proteins. When they make mucus, mucus has a lower density, so that total density actually reduces. So there is a double whammy. The mucus is suppressing net density and simultaneously acting as a drag line. But if we include clay, this improves. But the more important part is R square. This is where it gets nonlinear because the speed will increase dramatically if you could just make larger particles of the same density. The moment you do that, you get a really big win, and the bacteria, even though they're still working, cannot do damage, as much damage. I mean, again, remember, these are ecosystems. I shouldn't be using the word damage. They don't work for us. They're happy, they're doing their thing. From our point of view, it's suppressing. So that's the idea. Second question.
Second question, you were saying that the biggest need currently, or one of the really big ones, is buoyancy motors. And so I was wondering if there are projects that like take inspiration from, because we were also talking about like creatures that use
Biological.
chlorophyll and
Yeah, not yet. This is what I'm really excited about. We've been looking at squids for a while. So if anybody is interested in squid biology, there's some really cool chemistry. Squids do two things. They have a buoyancy engine, they can produce gas using biological processes, but they also make some amine compounds that are dramatically light. And nautilus and many cephalopods, for example, are completely neutrally buoyant. That's why energetically they use very little energy to go up and down. Uh, it'll happen that there are lots and lots of ideas in a biological space. We can talk about those when we sit down for the frugal oceanography discussions. But again, I think it's important to understand these processes before we were to ever engineer them. Uh, Another frugal science project, somebody came up with a temperature and salinity measurement device and mounted it on the head of a seal. So forget about the buoyancy engine. Seals go all the way to the bottom of the ocean, and they have now shown clean data of a dive and using these seals as I don't know how I feel about it, but if you ever see a seal with a little transmitter on its head and some probe, uh, some scientists are to blame, but they are now starting to think about a biological network because seals can go much deeper, much faster. I don't know. I think it's an interesting question. I have split opinions about it. Uh, does it do bad to the seal? Maybe the seal is totally fine. Biological sensors are an interesting space to think about. Whales, the impact is much, much, much less, but seals are more abundant. So anyway, I mean, it's wild. You have to think very broadly. Okay, Kamal and then Shivam. And then I know people have to leave, so don't worry about it. These discussions can digress. We'll do this for 5 more minutes, but if you have to leave, don't worry about it.
I
I
just
know
had
it
a
must be
question
too late. Yes.
around the video that you showed where you said it's snowing at the bottom of the ocean.
Yes.
And
Yes.
you said that this is biomass coming from the top. And I just was puzzled because there is all sorts of biomass at the top which has different shape, form, and so many things. But at the bottom, it looks all the same. So how do you make it?
Exactly. It's aggregation. When it all dies in macroscopic scale, it forms these fractals. So we've been studying how these fractals form. A whole bunch of cells with a ton of mucus glued together to form these aggregates that are fractal. And now, if you zoom in, very much like every snowflake, every particle is unique. Just like people have said that every snowflake that falls is actually unique. Let me just send a message. I just saw an urgent message. Please bring over full scope. Okay, sorry. Shivam.
Hello, am I audible?
Yes.
Yeah. Earlier I had one, now I have two questions. So one question, just a clarification, Professor. When you said, so could you explain again when the thing you drop and it goes, and how does it automatically come up for collecting the sediments? I didn't get that part.
That's a clever idea. That read the Notion paper.
Okay.
We had a trigger mechanism that essentially changes the, it's think of it as a belt or a mass, and there's a couple of ideas that we worked on. There's a really beautiful space in buoyancy engines, in how buoyancy engines are made. There are chemical reactions that can make gas, for example. So we tested that. So by the time it reaches the bottom, it hits the bottom. Uh, Inside the device, a chemical reaction happens that produces gases. And now gas phase has changed densities completely. That's used in buoyancy engines itself. There is electrochemical ways of generating gases. That's how buoyancy engines work sometimes, using very precise control. There's also another idea that you can leave your weight behind. You can leave, and again, it's littering, because every time you deploy it, you leave something behind. But kind of there are trade-offs associated with that too. So there's lots of ideas. Yeah. And I think we
One
worked
last,
on a particular one around gases.
I see. Yeah, one more question, Professor, which was from your last answer. Could you explain more about the fractals? Because I'm a bit interested on fractals. So what was the
Yeah, I
thing
think,
you?
one important thing to understand, first of all, why do they form fractals? Why don't they form nice spherical balls? Uh, One classic, if people are interested in the theory, you should read what is called diffusion limited aggregation. What happens in aggregation as a process is when you stick to each other, you generate new interfaces, but you also close down other places where you could add the particles. So in diffusion limited aggregation, if you make colloids, they will aggregate, but they form these star-like shapes because the paths to now arrive in these narrow passages of gaps that were created are too tortuous for randomly discovering. And that's why these sets of structures people had thought were quite porous. So this is another sort of an idea of porosity in these structures is important, because when you are fractal, you have a lot of surface area. Uh, Bacterial activity is very high. Your sedimentation slows down. But I'm giving, if you want to start in that space, I mean, we're just writing the theory paper on this aggregation of where the fractal dimension comes from. But you can start by reading, just if you search for the term diffusion limited aggregation, you'll find some beautiful experiments for how self-assembly can make fractal patterns.
Thank
Okay,
you, Professor.
we are almost out of time. Thank you so much for hanging out this late. Think about the ocean. We will see you on Friday. Make sure you bring an object that you are ready to break. Or, I mean, hopefully not break, but I can't guarantee it. Don't bring something too precious. So don't bring your, $3,000 SLR, and then it's like Manus told me that we're going to open this up. I'm not going to be